In this video, I will demonstrate the determination of human blood group using the slide agglutination method. The test involves mixing a blood sample with anti-A, anti-B, and anti-D sera to observe agglutination reactions and identify the blood type. The materials required for this test include a clean glass slide, anti-A, anti-B, and anti-D sera, blood sample, sterile lancet, dropper or mixing sticks, cotton and disinfectant, gloves for safety. Now place the clean glass slide on a label page or background where a B and our H symbols are clearly marked. This will help in identifying each reaction area easily while observing the agglutination. First, sterilize the fingertip using an alcohol swab to prevent any infection. Then, using a sterile lancet, gently prick the fingertip to draw a small drop of blood. Allow the blood drop to form and carefully place one drop in each of the labeled sections on the slide. Carefully place a single drop of blood onto each designated area of the slide, one drop on section A, one on section B, and one on near H or D section. Be gentle while placing the drops to avoid mixing or spilling. Begin by adding a drop of anti-A serum to the section labeled A. Then add a drop of anti-B serum to the section marked B. Finally, take the anti-D serum and place one drop in the section labeled RH or D. Make sure to do this step slowly and carefully, adding each reagent one by one to avoid contamination. Now take clean mixing sticks or toothpicks and gently mix the contents of each section on the slide. It is very important to use a different stick for each section to prevent the sera from mixing with one another, which could affect the accuracy of the test results. Finally, use a third clean stick to mix section A. Now we will observe each section of the slide carefully to check for agglutination, which appears as visible clumping in the mixture. In the results, there is no clumping or agglutination observed in section A, which means that antigens are absent. However, agglutination is clearly seen in section B and in the RH section. This indicates the presence of B antigens and a positive RH factor. Therefore, the determined blood group of this sample is B positive. You can clearly observe the agglutination by pausing the video. There is no visible change or clumping in section A, confirming the absence of antigens. On the other hand, noticeable agglutination is present in both section B and the RH section, indicating the presence of B antigens and a positive RH factor.